by introducing Anshul Kundaje. He's an associate professor in genetics and computer science at Stanford. I've known Anshul since he was a wee graduate student way back in the day. And he sort of first splashed onto the larger scene when he basically single-handedly did all the analysis for the ENCODE consortium and for the Roadmap Epigenomics Consortium. So he loves consortium science and is happy to talk about it at length, I'm sure. His group works on the kind of stuff that you see this title about, and you can read about, and he'll tell you all about it. But he's been someone that's been a pleasure to work with for a long time, and he's not quitting consortium science. He's also currently involved in IGVF. I know some people in our department are involved in that impact of genetic variation on function or something. That's what the F is. And he's also involved in a new consortium that I'm also involved in, Multi-Omics for Health and Disease. So all those things play into this overall goal of trying to figure out how gene regulation works. So take it away, Anshul.
Awesome. Thank you, Bill. And thank you for the kind invitation. So I'm going to talk to you today about how we try to use deep learning models with interpretation frameworks to decode non-coding DNA and gene regulation. Okay, I'm obsessed with sequence models. I try to map everything back to sequence and try to understand how sequence encodes function. And we're going to focus very specifically on regulatory DNA. These are, of course, elements scattered all over the genome. Their role is to regulate gene expression. They usually recruit, at least transcriptional regulatory elements, recruit DNA binding proteins, transcription factors. These transcription factors have evolved to recognize very specific sequences. And just like we have various kinds of logic encoded in coding sequences, there is complex syntax and grammar encoded in non-coding DNA as well. And so our goal is to try to understand what that syntax is and how it varies across cell types, cell states, individuals, species, and so forth. And our workhorse is to actually use biochemical profiling experiments across the genome, so sequencing-based experiments that try to profile binding of transcription factors, like your idea, this is encode data for a whole variety of TFs, transcription factors in one cell type, or other things like DNA-seq and attack-seq or single-cell attack-seq that measure chromatin accessibility or nascent transcription like ProCap. And given any such profile in some cell context of interest, we want to understand how DNA sequence is encoding or driving that activity. Okay? And as Bill mentioned, there have been large efforts like ENCODE, Roadmap Epigenomics, and then there are single cell versions of these now, HubMap and HTAN and so forth. But what you end up getting is large collections of molecular readouts across the entire genome in tens of thousands of cell states, cell types, single cells, individuals, and so forth. And so we mainly use machine learning models trained on this kind of data to decode regulatory DNA and then use these models in a counterfactual setting to predict effects of and predict effects of genetic variants. So the workhorse models I'm going to talk to you about today is these what we call base resolution models. These are models that are trained in a supervised fashion. Given some molecular profiling experiment across the genome that is regulated by DNA sequence, our models try to map sequence across the genome to base pair resolution profiles of molecular activity. And I'm a cheap person. I like to build cheap models. I wear free t-shirts and stuff like that. So the models I'm going to present to you are super cheap. You can train them in a few hours on a consumer GPU, and hopefully I can convince you that they can do things as amazing as Ferraris and Rolls Royces. So we built a series of these. It started off in 2021. This is a great collaboration with Ziga Avcek and Julia Zeitlinger. This is the BPNet model that we trained on chip EXO and chip Nexus data, which is high resolution transcription factor binding. Since then, we've adapted this to chip-seq and cut and run. We also have Chrome BPNet, which I'll talk a lot about today, which is adapted the same kind of model to chromatin accessibility profiles. We put out ProCapNet just recently, about a month or two ago, which is similar model trained to map sequences to nascent transcription like ProCap, CAGE, or steady state transcription like CAGE and Rampage. And we have upcoming models for other kinds of readouts from MPRAs, StarSeq, ATAC-StarSeq experiments as well. The cool thing is we use the exact same architectural backbone. We don't do a lot of architecture engineering. Our focus is on building very stable models that really model the effects of local sequence context. So we are currently not modeling effects of really long-range genomic effects. Our philosophy is that we first need to understand really how local sequence works. And if we figure that out, maybe we have a hope for building much larger chromosome scale or genome scale models. So I'll walk you through some examples of these models. But once we train these models, what do we do with them? We've built a series of interpretation tools over many years, one of which is we can take the model and ask it a question like, given this particular enhancer in the genome that drives this particular activity in cell type X, can you tell me which base pairs in that sequence are driving activity? So we have a series of tools like DeepLift, Yuzu, and FastISM that take a trained model, take its predictions on some query sequence and kind of reverse engineer contribution scores of individual bases to activity. So this allows us to query any individual sequence. Now, you may want to query tens of thousands of sequences and summarize the predictive patterns that the model learns. So we built TF Modisco, which takes these kinds of attribution or contribution score tracks across thousands of sequences through the lens of a model. The model claims that certain bases are important, certain are not. And that's what the height of these bases tell you. And we can kind of throw away the pieces of the sequence that the model claims not important, ending up with little seclets. You can cluster these and end up with kind of non-redundant motif representations. This is a nice way of summarizing the lexicon of patterns that the models have ended up learning. But then we want to not just sort of capture lexicons, but also higher order syntax. And so we can do really kind of cool in silico perturbation experiments. And here are some examples. You can think like a biologist and you can design a synthetic library. You can insert motifs or whatever of your choice. You can change spacing, orientation, affinity, and you can use a model like an oracle to predict effects. And so you can start graphing out the marginal effects of various syntactic properties of these sequences. Or you can act like a in silico CRISPR experiment, and you can take genomic sequences and you can, delete, change bases, whatever you want, right? And you can do this combinatorially on a very large scale, especially when you have a small model, you can do lots of experiments with it. And then, of course, the most natural perturbation experiment is to take genetic variants and push them through the model and see what it predicts, right? So this is the toolkit. It's very simple. I'm just going to use this toolkit across the talk now. Okay? So let's talk about BPNet. This was a simple model, again, mapping 2,000 base pairs of sequence to 1,000 base pair profiles of coverage at single base pair resolution. And the key idea is it's a very simple CNN, convolutional neural network, but it takes the input and predicts two types of outputs. One output is the total coverage over the entire sequence. So that's the sum of the reads coming from the experiment, a single scalar. And the other is a multinomial probability distribution that the model predicts as to how those read counts, the total coverage is distributed over all bases. That's the trick. So you break up the prediction into two components, the total magnitude and then the shape of the profile. So we had originally trained this model on chip nexus data, which is really high resolution base pair resolution data. We now have trained it on very large amounts of chip-seq data sets, which are much lower resolution, much crappier data sets capturing transcription factor binding. And I want to show you some really cool results we recently have, just showing forget about the biology, just what these models can do in terms of data cleanup and denoising, right? So here's two chip-seq data sets for the same transcription factor E2F6. This one is the higher quality replicate, if this looks good to you. This is the lower quality replicate. This data set has 3 times the number of peaks as this one. They look completely different. There's very little overlap. But now we train separate models on each of them, okay, the BPNet models. And this is the prediction that the two separate models make for that exact same locus. You see, first of all, beautiful imputation of the profiles. I mean, this is derived from this, which is quite amazing. Again, these are test sequences, just to be clear. We're not training on this locus. And so you can see that the two replicates suddenly start looking a lot more similar. And if we actually not just look at the predictions, but look at the underlying sequence drivers that the model is claiming, they are bang on top of each other. There's an E2F6 motif and a MAX motif collectively driving binding at this location. And you can, of course, do this genome-wide, and you can compare the Jensen-Shannon distance between the observed profiles, that's this distribution across all the peaks. You can see it's really bad. The worst GSD would be 1, and that's close to where we are. If you look at these two, that's pretty much what it looks like. This is what you get after you take the predictions from the model and you compare them head to head. So you get this amazing dramatic improvement. And this is not for one cherry pick data set. We have literally hundreds where they've done ChIP-seq or, from different antibodies, some done 3 years earlier with a different sequencing technology. And across the board, you get really beautiful improvements in concordance of these experiments. So just that is a big benefit, that you get a lot of beautiful denoising and imputation. But we can do cooler stuff, like we can actually explore syntax, as I said. So here's one example of a FOXA ChIP-seq dataset. FOXA2 is an interesting pioneer transcription factor in liver and HepG2 cells. And when we train a BPNet model and apply TF Modisco to it to learn motifs, it learns standard motifs like FOXA, HNF4, and CBP. All of these 3 transcription factors are very interesting. They're all potentially pioneer factors which can open chromatin by themselves. They're supposed to act very independently or regulate binding of other TFs. But what we see is very interesting. We can do these in silico syntax experiments where we're inserting a FOXA motif and a CBP motif next to each other. And all we're doing is changing the spacing between them. And we're graphing the increase or the change in binding of FOXA2 as predicted by the model as a function of this spacing. And you can see this pretty dramatic cooperative binding as you start moving this CBP motif towards the FOXA motif. You see this kind of really nice exponential rise in binding. And here's sort of an animation of what that looks like. And if you just snapshot this, you can see even up to 200 base pairs apart, these two motifs have a pretty interesting cooperative effect. Now you can do that for HNF4 and FOXA, 2 other motifs, right? Again, with respect to the FOXA2 binding, and you see a different graph, right? So these are all soft syntaxes. These are not hard syntaxes. It's not like the 2 motifs have to have an exact spacing. It's very flexible. And there are rules. I mean, there's sort of a transfer function that as you change spacing, the model, cooperative effect changes as a function of distance. We can do similar things for orientation, for affinity. Very cool. So you can do a lot of this. And using this kind of approach, we've now trained models for tens of thousands of data sets from ENCODE. And I have to say the syntax map of the human genome is amazing. It is just insanely complicated. There's hard syntax, soft syntax, different transfer functions. This is just pairwise. Now you can imagine what happens when you take higher order combinations, right? The amazing thing is the models are able to learn this quite effectively, and we have validated many of these things in experiments previously. So what I want to talk to you about next is a very interesting phenomenon of deep learning models are these kinds of black boxes. You train them on read count data. They can make some cool predictions, but can we link them back to biophysics? Like, transcription factors have been studied for years, but from the perspective of affinity and synergy and kinetics and so forth. And it's always been intriguing that if you take in vitro experiments that directly measure thermodynamic affinities, like in this case, a BET-seq experiment, and this is done on the 4,4 transcription factor in yeast. There's a large library of sequences where we take this E-box motif of this 4,4 transcription factor, which is CACGTG. We keep that core motif constant and we simply modify the flanks. So we're taking 5 base pairs to the left and 5 base pairs to the right, and we're keeping the core motif the same, and we're changing the flanking sequences. And that actually has a massive impact on affinity of this particular transcription factor. And we can measure that very accurately with this BET-seq experiment. Now, for that same transcription factor, you can do an in vivo experiment in yeast cells with ChIP Nexus or ChIP-seq. And you can take the exact same sequences that you see here in this library. You can find them in the genome. You can often find multiple instances. And you can just say, what's the correlation between the ChIP-seq readout, the in vivo occupancy measurement, and the in vitro affinity, right? It's often really quite poor. So in this case, we're looking at ChIP-seq here on the x-axis. On the y-axis, on the x-axis is the measured delta G's, affinities. And you can see that the correlation is really quite poor. It's close to 0.4. It's inverted because that's how it should be. So there's always been the discrepancy, and often it's worse than this. This is in yeast. If you go to mammals, as I'll show you in a few slides, it gets even worse, right? So this weird discrepancy between what we see in terms of in vitro affinity and in vivo binding. So we came up with a simple way of trying to marginalize out. If you think about what a BPNet model is doing when it's trained on ChIP-seq data in the genome, it's probably learning affinity of individual transcription factors, but then it's learning a lot of other stuff that's happening in chromatin, multiple TFs binding together, syntax, and so forth. And so we thought that maybe there's a way to simply do a simple way of marginalizing out all the other effects and only sort of distilling out the effect of an individual chimer, right? How would you do that? Like literally marginalization, you would integrate out all the other effects. So the simple idea here is we can, take this, let's say this is the chimer that we're interested in. We want its affinity. We can take this chimer and shove it into some random background. And then we'll take the BPNet model and we'll predict it's the trained BPNet model that's trained on ChIP-seq, and we'll predict its effect from the random background sequence and the random background sequence with the chimer in it, right? And then we do this for a very large number of background sequences. We just compute the log fold change of this, the sequence with the chimer versus the sequence without the chimer. And then we average over all the backgrounds. So we're basically integrating out the effects of all the background bases except that chimer, right? We call this affinity distillation, fancy word, but it's just like averaging background effects out. Now see what happens, right? This is the same plot I showed you before. This is if you take the actual ChIP-seq data and compare it to the DDGs from the in vitro experiment. Now we have a BPNet model that's trained on this exact same ChIP-seq data, but now we're using the marginalization trick to try to predict the marginal effects of these schemers. And you see this dramatic improvement comes to 0.81, right? So this is really cool because it shows that the models in fact are likely learning something like affinity. It's not calibrated, but at least it's highly linearly correlated with affinity. And these are sequences that are in the genome, right? That's why it's a small number, but the library has millions of sequences in it. Most of them, those are not in the genome, which is why there is no point here, right? Because you don't have a ChIP-seq, observed ChIP-seq point for that sequence. But then we can predict for all these other sequences that are not in the genome at all, and the correlation pretty much stays the same. So you can extrapolate out of distribution, well, not out of distribution, I guess it's still interpolating, but these are sequences that are not seen in the genome, and you can predict affinities really quite effectively. This is in yeast. So does this actually work in mammals, in humans? And it surprisingly does. So here we're looking at MYC in HeLa S3 cells, and this is the correlation between, again, the observed ChIP-seq data and the surrogate affinity as measured by protein binding microarrays. So we actually changed the technology now. Rather than BET-seq experiments, we're looking at PBMs. And again, you can see the actual correlation between this is terrible. It's like 0.18. But now as soon as you do the marginalization trick, you get this amazing 0.77 correlation, right? So even in humans, it actually works out really nicely. Now, MYC binds very differently in HeLa S3 cells and in HepG2 cells. The in vivo binding patterns are very different. Even though they bind the same core motif, they cooperate with very different factors. But the affinity of the transcription factor should be the same, right? So the hope would be that if you train the ChIP-seq dataset, a BPNet model on ChIP-seq of MYC in HeLa, and you instead trained it in K562, which is a different cell line, right? Even though the in vivo binding patterns are very different, if this claim that we're making is that the model is learning affinity, the marginalization should work equally well for both. And that's what we see. So this is from HeLa S3, this is from K562, very similar results, right? You should check out the paper. We have a lot more stuff in there. We even are able to distinguish between factors of the same family. We can train ChIP-seq data on two factors like FOFO, CBF1, various BHLH factors, and you can beautifully, from the in vivo model, distinguish very subtle differences in affinity between factors of the same family. So the ChIP-seq models actually are learning a lot besides just some black box neural network. There's biophysics sort of built in there, and this opens up new avenues to potentially train models jointly on in vitro and in vivo data, right? Which is going to be very exciting. There's a lot more in vitro data. It's often cleaner. It captures only one aspect of binding. All right, so let's move to chromatin accessibility. So Chrome BPNet, we just added 4 more letters because actually this model is exactly the same as BPNet. All we do is increase the capacity of the model. The architecture is identical. We go from 64 filters in the first layer or in the layers to 512 filters because we need to capture binding of many transcription factors. This model also works well. The quantitative correlations of total accessibility across peak regions is quite high. Also, if you look at the Jensen-Shannon distance between observed and predicted profiles, they are very close to replicate concordance, much further from baselines like randomly shuffled profiles or the average profile. And if you look at the genome, you can actually see that. You can see this is the observed data at base pair resolution. This is the predicted data at base pair resolution. They're really indistinguishable. But unlike the ChIP-seq data models where we interpreted them, and you saw these beautiful motifs pop up, for attack-seq data sets, when we train these chrome-dependent models on attack-seq, we not only see motifs like right here, this is a CDCF motif, but we see a whole bunch of other garbage lighting up all over the place. It's like the attributions are super noisy. So initially we thought, oh, maybe the models are noisy. But then, we try to use Modisco to summarize what the patterns are across the entire genome. And what we realize is the models are actually learning enzyme bias. So the motifs that really dominate the model are not motifs of transcription factors like RUNX and IRF, which they should be. It's these TN5 bias motifs. So TN5 is the enzyme that attack-seq uses to tagment the genome to perform this experiment. And TN5, the enzyme that's used, has its own sequence preference. If you train a model, very powerful neural network on attack-seq data without worrying about any biases and so forth, it ends up learning biases. It spends all its time, a large amount of its parameters learning biases, and the motifs kind of get sidelined, which is why if you go back to this slide, here's a CDCF motif and all this other crap right here, this is likely just enzyme bias, okay? It also showcases why it's very important to interpret models, because if you just look at prediction performance, we would stop right here, right? We'd be happy, we have solved the problem and go home, but the model has actually learned not just the biology, but lots of other artifacts in the data. The same story we see with deep learning in all domains, right? So how do we correct this? Well, there's a nice trick that we get. The human genome is massive and it's full of junk, which is a bad thing and a good thing. It's a good thing because you have 90% of the genome actually consisting of regions which are not accessible, background regions. So you have a control experiment for free, right? You have all these other base pairs in the genome where transcription factors are not binding. The only thing that's hitting there is TN5. So if you wanted to learn a good model of TN5 bias, you have a beautiful training set. It's everything that's not attack-seq peaks, the rest of the genome. That's what we do. We just train a neural network on the rest of the genome, not the attack-seq peaks, not the chromatin accessible sites. We train this model, and when we interpret it, we see that it learns TN5 motifs, a beautiful model of TN5's enzyme bias. Now what we can do is we can retrain this model where we first take this frozen bias model, we fit it to the profiles in peaks and background. So we do basically try to explain as much of the signal with the bias model. And then we fit a second model that learns the residual, that is the portion of the signal that cannot be predicted by the bias. Now, once you train this model, you can disconnect the bias model, and what should remain is the clean residual, right? The stuff that is not biased. So that's exactly what happens. So this is the same region I showed you before. Now you can predict the bias in that peak region because we had trained a bias model. Notice how similar the bias is to the observed profiles, except right here, where CDCF actually, the motif, the transcription factor is blocking tagmentation in the actual attack-seq, but in the bias model, you can see that there's quite a bit of cutting going on. You can interpret the bias model, and you can see that many of these spurious bases that we were seeing before are very similar to what the bias model learns. And then we can regress out this bias and look at the residual, and there's this beautiful latent footprint hiding behind this data. And here's a CDCF motif. All the spurious bases are gone away, and there are some little ZBTV7A motifs lying around next door as well. If you try doing this with other bias correction methods, ad hoc correction methods, they don't actually work as well. These are two other methods that have been built in the past. You can see the biases remain, even though they kind of partially correct for effects. So you can use the power of the neural network, which is very good at learning biases, to only learn biases, then regress the bias out, and then use another powerful neural network to learn what is not bias, and then it learns really nice biology, right? And we have proof because we can not just look at one example, we can now run Modisco on the bias-corrected contribution scores across the entire genome, and all that remains is beautiful motifs. So we've eliminated bias quite effectively. Now, we can train such models on two different assays, ATAC-seq and DNA-seq, which both try to basically capture chromatin accessibility. At low resolution, they look very similar. Here's an ATAC-seq peak when you smooth it out. This is what it looks like at base pair resolution. If you train an uncorrected model, just a model without bias correction on this data, you can see it looks beautiful and very similar to the observed data. We can train a similar model on DNA-seq this time that also measures chromatin accessibility, but using a different enzyme, right? The DNA is one enzyme. And in this case, you can see that even though the peaks look very similar at low resolution, the base pair resolution profiles for DNA-seq and ATAC-seq look extremely different. And again, if you fit neural networks to either of these data sets naively, they copy the data exactly. They exactly learn how sequence drives those profiles. But once you do bias correction, you can see this dramatic change in the profile. Again, you see now this kind of really nice footprints showing up, and here's the underlying sequence syntax that the model predicts. And if you do the same thing for the DNA-seq data set, you see these footprints lining up very nicely and the exact same syntax being picked up by the model. So you can train the model now on two entirely different data sets with very different biases. But once you do bias correction, at least on the footprinting, they align quite nicely, although DNA-seq gives you much sharper footprints. It is a smaller enzyme, can go in there and cut much better. But the underlying sequence syntax, the two models learn are nearly identical. And again, you can verify this genome-wide because this is the similarity in the profiles before bias correction. This is if you take the predicted profiles without bias correction, you can already see some improvement because of the denoising. But once you do bias correction, the footprinting lines up much better. So you see this amazing reduction in the Jensen-Shannon distance between two separate experiments, right? Similar story to the replicates I showed you for the ChIP-seq data sets. These are two different data sets from different enzymatic reactions to measure chromatin accessibility. And it's not just for the predicted profiles, but if you look at the contribution scores, right, the syntax that the model is predicting, that also shows a dramatic improvement after bias correction. Okay, so this hopefully proves that the bias correction helps really learn just the biological component, helps align different assays together. Now, what can we do with this? It's very cool. So we've applied this recently to a very large multi-ohm atlas of human fetal tissues. And we have 203 cell types, and we've actually trained models on all of these. And now we try to summarize the entire lexicon, like what are all the motifs that drive chromatin accessibility in 203 different cell types in various human fetal tissues. And it's pretty amazing. The lexicon is quite large. We learned 525 unique motifs. Most of them are actually not base motifs. So they're not motifs of individual transcription factors. Only 92 motifs of individual transcription factors are learned that drive accessibility in all the 205 cell types. But look at the composite motifs, that is combinations of homotypic or heterotypic units of transcription factors, very large collection of heterocomposites, 224, a pretty large number of homocomposites as well, mostly activators, some repressors, right? So there's a very complex lexicon that's not just consisting of individual transcription factor motifs, but complex composites where you see combinations like EBOX and EBOX or GATA and MEF with very specific fixed spacing syntax. I just want to show you one example of what this looks like. These are all the various heterotypic units that we see for BHLH and homeodomain composites. You can see kind of really cool, all different kinds of combinations with different spacing. And we learned so many interesting motifs that only have been recently discovered through painstaking experiments. So there's a very famous coordinator motif that was discovered by Joanna Weissoker's lab just this year and through very complex experiments. And this just shows up right there. It's the exact same motif that we find. And many of these are super cell type and tissue specific, right? So you can really start tearing apart the motif lexicon, which is much more complex than the motifs that are in current databases. These monomeric, motifs or transcription factors, they don't act by themselves. They often act combinatorially. And we just looking even at chromatin accessibility, most transcription factors don't drive chromatin accessibility. But even if you take just the TFs that drive accessibility, it's still a very, very large lexicon of these homo and hetero composites that we're now starting to be able to discover. So what else can we do with it? We can, of course, use a model, as I said, for variant effect prediction. Very simple. You take the reference allele, alternate allele, push it through the model. The model predicts profiles, and then you can look at the allelic impact. But you can also interpret the model and see what's going on. So here you can clearly see that this variant, the C to the G allele, when you swap the C to a G allele, it creates a very strong SPY1 motif, and these two SPY1 motifs together drive accessibility of this enhancer, right? So you have a nice explanation for the effect of the variant as well. So we try to benchmark this on a large scale. We start off with a very nice QTL data set that Steven's lab recently published. This is a QTL data set measuring attack-seq in lymphoblastoid cell lines across 100 diverse African individuals of African ancestry. And we were able to discover about 11,098 significant CAQTLs. So we can take these CAQTLs and we can, our model is trained on one reference data set. It's a European LCL, so it's from a different ancestry group. And we're trying to train one model on reference genome on a reference individual and trying to see if we can predict through counterfactual predictions the effects of all these variants in an African cohort. And actually, the predictions are quite decent. You don't see much directional discordance. You see the quantitative correlations are not that great. But to be honest, if you take two different QTL data sets and you compare their correlations, they are about the same. They're slightly higher, maybe in the range of 0.8, 0.82. So there's still a gap. But do remember that these are local sequence models. We're only modeling 2,000 base pairs of sequence. So it's kind of crazy that the model has never seen genetic variation, is modeling only local sequence, doesn't have any directional discordance, and actually has a pretty decent correlation, right? So we're going somewhere. We're not completely in the woods. What's very interesting is we benchmarked against Informer, which is a much bigger model. Informer is this big multitask neural network, which models about, I think, 200 kilobases of sequence. It's trained on thousands of ENCODE and roadmap data sets simultaneously in human and mouse. It's a big transformer model. So in principle, it should do much better than us, right? But actually what we see is two things. Firstly, if we take the scores published in the Informer paper, they're abysmal compared to us. So these are our models, and this is just, we try to do threshold-free analysis. So like you can always pick a threshold where your model looks good. So all we do is we change the p-value thresholds for the QTLs, and we try to model how performance changes as a function of threshold. So you can see across the board, the performance is pretty strong for Chrome BPNet. And Informer does surprisingly poorly. Now, this did not make sense to us. It should not happen, right? So we actually realized that they had a mistake in the way they were calculating scores. Since they are a long context model, they predict profiles at over entire 100 kilobases, right? So when they predict the effect of a variant, they take the prediction from the reference and the alternate allele over the entire 100 kilobases, and they do a log fold change or a difference. Now you can imagine if there's a peak sitting in there that changes and the rest of the genome does not, it'll just get swamped out, right? So all we do is we replace it with the way we compute it, which is locally, a local effect. And then you can see Informer's performance improves substantially. It's also a good lesson why DeepMind should release their models, because if we didn't have access to their models, we would not find the bug in their code, right? But even after we fix the issue with their model, you actually realize that Chrome BPNet is in fact much better than Informer, trained on the exact same data, right? Even for binary prediction, so this was quantitative prediction of effect sizes on the significant QTLs. Another interesting question is, if you just scored all the variants in the genome, could you discriminate the significant QTLs from the non-significant ones, right? So that's more of a classification task. And we can again look at performance. This is what a random predictor would look like. It's a very highly class imbalanced problem. You have 11,000 significant QTLs and tens of millions of non-significant ones, right? So this is the baseline AUPR that a random classifier would have. This is again Informer using their original scores. This is Informer using their revised scores. Again, you can see Chrome BPNet models trained on the exact same data set have very comparable performance, but here's a trick. Simply increase the sequencing depth of your training set, okay? And you get much better performance. You don't need a big model, you need more data. That can actually help you get better performance than training a bigger model. There's recent evidence that if you further augment this with personal genomes, maybe 20 to 30 personal genomes with corresponding profiling data, you can further improve this performance with a small model, not with a big model, right? So there are tricks you can do without worrying about model size and scale and so forth, which can get you pretty far. And then, of course, you need the scale, you need transformers, do need models that can scale the entire genome. But I think it's important to think about how to build these models piecewise and really understand where they fail and figure out simple tricks like improving data quality, improving read depth, which can give you a big bang for the buck. We've then benchmarked models on not just chromatin accessibility QTLs, but one important use case is GWAS fine mapping. So here we've looked at, this is GWAS from various cardiac traits, and here are posterior probabilities of fine mapping of variants. So as we increase the posterior probability of fine mapping, we see that the enrichment of Chrome BPNet scores with respect to the fine map variants dramatically increases. You can see up to 100-fold enrichments. This is telling us that the variants that Chrome BPNet is prioritizing also are prioritized by a GWAS fine mapping method. So we're seeing kind of nice enrichments for fine mapping of GWAS. I'll skip this in the interest of time, but there are some really cool like allelic interactions of variants that occur in the same enhancer. You can see really interesting cooperative effects. But I also want to point out quickly that we can also do rare disease variants, not just common disease and GWAS. This is a very cool case study where this patient actually has a rare thyroid disease. We took this variant and screened it through almost 1,000 models that we have, or 3,000 models that we have, spanning a whole variety of different cell types and tissues. There is only one model that scores this variant as having a significant effect, and that is a model trained on ATAC-seq in thyroid. Okay, so the cell type specificity is bang on, and the mechanism is super interesting. You can see that the variant is in fact in this STR element, and when you mutate this base, it's a single nucleotide variant. We mutate this T to a G, it doesn't do anything to the proximal motif, but you can see this Fox motif pop up next door, and there's a massive cooperative effect on all the motifs next door. So you have this really cryptic effect that's going on, which you can only interpret with the help of the model. And this was recently validated with actually single molecule fiber-seq data in this recent paper. Okay, so we even have independent validation of this effect. So just to summarize, hopefully I showed you that neural networks can learn accurate models of context-specific regulatory activity. The models, even though they are black boxes, they can be interpreted and you can decipher novel motifs, complex syntax. You can distill the thermodynamic affinity from these. The assay biases can be detected, learned, and corrected, improving concordance. You can impute missing signal and reveal causal biological drivers. You can predict counterfactual effects of variants on chromatin accessibility, report expression. You can fine map GWAS variants and even do rare disease variants. And small models can often outperform. So be cheap like me. And with that, I'll stop and just thank my lab and all the lab members who helped with this.